How do bacteria adapt to salinity? We have not been the first ones, and unlikely are we the last ones to address this broad question. However, recently, we have made a major step forward by identifying common evolutionary dynamics and convergent adaptation strategies via a systematic analysis across the whole bacterial tree of life. These efforts have been published last year in Science Advances. You can access the publication via the QR code or by clicking at it. It should get you to the publication website. I will not do it now. What I will do, using this poster, I will more briefly sum up the study. My name is Krzysztof Jurdziński. I'm a PhD student at Anders Andersson's lab at KTH in Stockholm, and I have co-led this study together with Malika Kmershek. The main premise here is the idea of three aquatic biomes defined by salinity, freshwater, brackish, and marine, with distinct bacterial communities inhabiting them. We not only use this idea from previous research, but we further corroborate that this is actually the case. Then to perform our comparative analysis, we've collected publicly available metagenome assembled genomes from the three biomes. Big thanks to everybody who contributed to collecting the samples and generating the data and assembling the metagenome assembled genomes. After quality filtering, we got more than 11,000 marks. We then clustered them into 95% average nucleotide identity clusters, and we got more than 3,500 of what I will from now on call species. We reconstructed the phylogeny, and then we identified most recent transitions. We leveraged the wide phylogenomics scope of the study, and we went for relatively assumption-free way to identify transitions. And that is by looking at pairs of what we call monobiomic sister groups. A monobiomic sister group is a monophyletic group with representatives from only one biome, with a sister group with representatives also from only one biome, but from a different biome. So the ancestral nodes, there had to be some transition one way or another. We might miss some of the transitions, but we'll get a big number of high confidence transitions across the phylogenetic tree of life. If we identify them that way, and that's what we've done, we looked at the dynamics, so at the frequency, direction, and the estimated time when they happened, and then we used them to do comparative genomics on the closest relatives from different biomes. So we looked at in-fed proteomes and we looked at gene content. So heading to the results, the first part, as I mentioned, is about further looking at the separation between the biomes, and we set it at the species level. Here I show just the numbers of species which were unique to the biomes. That's been at least hundreds, while none of the species were found in all of the biomes, but a few had representative among the marks from different biomes. And that's been despite the fact that we had marks coming from very closely located samples from very similar climatic conditions, but from different biomes. We also looked at within the brackish biome at two different bases. We had the Baltic and the Caspian, and those, despite being geographically isolated and having quite different conditions, shared much more species. And also those were two different data sets. However, looking at the shared brackish species, we could see that the populations of those within those species have been the strongest structuring factor has been the isolation between the brackish, the Baltic and the Caspian seas, and much less the effect of pronounced environmental gradients of the Baltic Sea, suggesting that it might be a geographic barrier that affects population structures. Furthermore, we identified the transitions. There were quite rare events, and we've also estimated the time since they happened. And here, according to our estimates, it's been at least millions of years for most of the events. Maybe even more strikingly, even if our estimates are orders of magnitudes wrong, we do not see any pattern that could correspond to the big geological events which were Baltic Sea and Caspian Sea formation, suggesting that after they've happened and after the local environment changed from freshwater or marine to brackish, it was rather migration than local adaptation that filled in the niches. Finally, we've also looked at the direction of the transitions, and for most single events, we could not assign a direction with high confidence. However, looking at the probabilities across all of the events, we could see that in general, it's been much more probable that most of the events happened from marine or freshwater into the brackish biome and not out of it. Then we moved on to comparative analysis, leveraging our definition of transitions, which were pairs of closest relatives from different biomes. We compared the genomes of those closest relatives. We've looked at inferred proteomes. And here, following Cabello-Yebes and Rodrigo Valera, we've seen that there were less of neutral proteins in higher salinity, and there were also less acidic proteins in freshwater bacteria than in the counterparts from other biomes. We went further with those results also. I didn't show it here, but we also show that these changes most likely continue to happen, those shifts in proteome properties, long after the transitions, according to our estimates, tens of millions of years. And this fitness mismatch after transitions might be one of the barriers that make them such a rare events. And we've also connected those transitions to those changes in isoelectric point distribution to composition of inferred proteomes. So in higher salinity, there was, of course, more charged proteins, but maybe more strikingly among those, it was rather the proportion of acidic than basic residues that increased. And we also look at specific proteins, specific residues more in detail in our paper. Then we've looked at changes in gene content and gains and losses of specific gene functions. And here I will not go through all of the genes. There are quite a few. I here only exemplify the results for one transition type. We've looked at genes that have been consistently gained or lost during the same type of transitions by diverse groups of bacteria. And we got some genes that have been discussed in context of adaptation to salinity, like osmoregulators or any coupled transport. Other that haven't been that much discussed but make a lot of sense, like magnesium transporters, and others which role is much less obvious or unknown. And here maybe the best example is a putative membrane protein, for which we just don't know the function now. So to conclude, bacteria adapt to salinity, to a new salinity regime rarely and over long periods of time. While they do it, they undergo wide scale, changes in proteome properties and composition, and they gain or lose specific gene functions. And those gains and losses might allow them to transition despite the challenges connected to adjusting their proteome properties and composition. Thank you very much for your attention, and I wish you a wonderful conference.